All right, this simple animation, or it's not an animation, this simple video is a drawing, a simple drawing of intracellular digestion, which is something that some cells do. And it's an important pathway or process in the cell. And in this case, the cell is going to take in something from the outside environment. So that's shown here in orange and black. It could be a mixture of things, macromolecules, dead cells, even live cells, if those cells are small. And it's going to take in this food particle, so to speak, by a process called endocytosis, and specifically called phagocytosis. So there's going to be a little indentation where the food particle is going to be brought in. We can move that food over to where it's over here. And then the membrane is going to surround it and bring it into the cell and pinch off a vesicle inside the cell. And that's called endocytosis in general, and specifically if it's bringing food particles for digestion, it's called phagocytosis. Now, once that vesicle is in the cell, we call it a food vacuole, but it's a type of vesicle. And there's another organelle that is going to be involved, and that's called the lysosome. The lysosome is shown in darker green, and the lysosome has enzymes in it that can do hydrolysis of macromolecules, hydrolysis of polymers. And you'll remember that hydrolysis of polymers produces monomers. And so when these two things fuse together, these two membrane-bound compartments fuse together, you get a larger compartment, I guess. It would have to be larger, where the two have fused together. And that's going to mix the hydrolytic enzymes with the food particles. If these are live cells, they will be then killed by this process. But basically all the macromolecules are going to be broken down. All the polymers are going to be broken down into the monomer units. And the monomers will be allowed to pass over the membrane into the cytosol. So that's the absorption of those monomers. Once the polymers are broken down into monomers, the monomers are passed into the cytosol. So maybe I should show those as little dots. Some black monomers and some orange monomers that end up in the cytosol. Anything that cannot be broken down, anything that's left in this vesicle, I'll draw it the same for continuity. Anything that's left in here, maybe there's a few little things that couldn't be broken down. Anything that's left inside of the vesicle is then moved back to the membrane, and the membrane fuses with the membrane. And then those waste products will be dumped. So this whole process begins with a phagocytosis on the far right of the screen. Phagocytosis, that brings the large macromolecules or even entire cells into the larger cell. Then the fusion of the lysosome with the food vacuole. And then the breakdown, the hydrolytic breakdown of the macromolecules and the absorption of the monomers. And then the release of wastes, which is this last step. It's an exocytosis, and it's also called excretion, release of waste. So phagocytosis is a type of endocytosis, and excretion is a type of exocytosis. So this entire process is how cells individually do digestion. We call it intracellular digestion. But they're really the three phases: the phagocytosis, the lysosome fusion with the food vacuole, maybe 4 steps, and the monomer absorption, and then the excretion of wastes, intracellular digestion process. And you'll notice in this one, even though I drew the nucleus and I drew the ER with the ribosomes, those were not involved in this process, not directly.